Sri Gurubhyo Namaha. Welcome to your online learning initiative and I welcome you all for the biology class. In last class, we saw about multiple alleles. An example, ABO blood group. I told that I'll give you a small question to check out whether you can do the cross. So we'll move to the question. So here is a question. If in F1 progeny, okay, all the four phenotypes are possible, like A, B, AB, and O. So what will be the genotype of their parents? So it's a tricky question, but very simple question. I'll read and I'll just pull out the keys, okay, pull out the clues. Always the question will carry a clue. We have to just check that. So we'll see here. So in F1 progeny, okay, all the four phenotypes of the blood groups are possible. So we'll make a note the four phenotypes A, B, AB, and O. All the four phenotypes are possible. Okay, so this is the clue. We have to pull out the genotypes of their parents. Okay, the clue is all the four. And now we have to think about the dominant and recessive. So here, what are the dominants? A, B, they both are dominant. The dominant when they express when they are in homozygous condition. So here, if it is A blood group, okay, when they are in homozygous, it will express A. It can also be expressed in a heterozygous condition, right? Then B blood group, when they are in homozygous as well as in heterozygous, they can be expressed. Okay, now this part is ruled out because here there are no possibility of O blood group. Either there will be A or AB only, no O blood group. So this part is ruled out. Okay, so here it is O. Okay, so we have to select this two only. You may ask me, why can't we select O blood group? We can't say O blood group because when you take O, either A, B, or O, only these three you will get. A, B possibility will not be there. So we will select these two and we'll check out. So the parents, IA, IO, IB, IO. Just indicate with the symbol male, female, right? And now pull out the gametes. It is not necessary here. You have to pull out the gametes, then F1 generation on selfing. Okay, directly we can go to the Punnett square. So here we'll go to the Punnett square. Gametes we all know. If you want, we'll write and complete that too. IA, IO. Here IB, IO. Okay, and circle the gametes. Right? Now we'll move to the Punnett square. So draw a box and show the Punnett square. Sorry. Okay. Here, just draw male and then female. Okay. We'll change the color. IA, IO. Male we kept as IA, IO. So right. And female, IB, IO. Okay. Now bring it together. IA, IB. Both you have to write I, not one I, AB. Okay. It is an allele. So IA, IB. Then here, IB, IO. Okay. And here, IA, IO. And here, IO, IO. So, we have all the four blood groups. AB, right? Then B, then A, and then O. So, it's very simple. Okay. Now, we'll move to the next topic. It is about polygenic inheritance. Always, as I said, from the word will describe it. What is poly? Poly is nothing but many. Genic is nothing but genes. Many alleles, okay, here they are going to involve to express one particular character. For example, if it is a skin color, if you take it, the skin color we are denoting with the three gene A, B, and C. Okay, don't confuse children. ABO is blood group, and here we are denoting the genes as A, B, and C for the skin color. So we'll see one after the other. Here, yes, it is a type of inheritance. Which one? This polygenic is a type of inheritance. Poly, keep in mind, it is many, in which traits are controlled by three or more genes. Three or more genes. Okay, where are they located? Whether they're located in the same locus or different locus. It will be in the same locus. Okay. The phenotypes reflects contribution of each allele and is also influenced by the environment. Okay, even environment plays a role here when it comes to skin color. Okay, UV rays also plays a role here. That's what we explained here. So we'll see an example. It will be still more clear for you. For example, skin color, human skin color, three genes, as I said, A, B, and C. Okay, I'm using the word gene here, not allele. When we are writing in a pair, we call it as allele, right? And the recessive, always when we start with the dominant, we'll talk about the recessive also. So capital A, capital B, capital C, dominant, small a, small b, small c, recessive. So here, so we see the example. It is nothing but the skin color. You can see different types, shades of skin color, right? So still more, we'll complete the alleles and then we'll go to the next. A, B, C, capital A, capital A, capital B, capital B, capital C, capital C. Okay, this is dominant. Small a, small b, small c, recessive. Now, when it is intercrossed, okay, what will be the genotypes, right? If you see capital A, capital B, small b, capital C, small c. Okay, so this is intermediate skin color. Okay, intermediate skin color. And what about this skin color? It is dominant black or dark, and here light shade, okay, or fair, okay, or white. I'll write in, since I've written black, I'll write the opposite, white. Okay, so black, the dominant, capital A, capital A, capital B, capital B, capital C, capital C, and white, recessive, all in lower case, small letter. When it is intercross, you can see the intermediate color. It is in a heterozygous condition, right? Even this we can talk about trihybrid cross. Yes, but here why we are not stressing the term trihybrid cross? Because here we are talking about all the three genes saying one particular character. It is nothing but skin color. That you keep in mind. Okay. Next, pleiotrophy. Pleiotrophy, it's something both more or less somewhat same only, but here it is a different totally. In polygenic, many genes, okay, will affect a particular character, one character only, okay. That is nothing but the color. But here pleiotrophy, multiple effect of single gene, one gene alone, okay, will affect many characters, okay, many traits like color, shape, size, like that it may affect. Okay, again I'll show you this slide. Polygenic, many genes, okay, one particular character alone they will control. So here there are three genes, they are controlling one particular character. It is nothing but color. Okay, skin color, the trait. Here if you take pleiotrophy, multiple effect of a single gene. Okay, so it is a phenomenon in which single gene, one gene alone, the gene may be TT or the gene WW or anything, one particular gene alone will cause many effect in traits, many characters, color, shape, size, etc. Right? Exhibits multiple phenotypic expression. The pleiotrophic genes affect the metabolic pathway, resulting in different phenotypes. So we'll see an example. Phenylketonuria. Okay, it is an example. I'll show you. Yeah. So it is caused by, always that should be a major cause. The major cause is nothing but mutation in the gene coding for the enzyme phenylalanine hydroxylase. Okay, so I'll give you one example. Okay, actually, the enzymes will have a code. Okay, our body is full of codons, right? If you see, numerous codons will be there. That we'll see in next lesson. So one example I will say, there are codons we saw in last class like A, T, C, G, adenine, guanine, cytosine, thymine. Like this codes will be there. So triplet codon. We'll take a triplet codon like this. Adenine, cyanine, cyanine, cytosine, cytosine. Okay. This codon, due to mutation, one particular thing. Okay, you can see one particular code alone, it was changed. Okay, it's changed into G like this. Can you see here the change? Yes. So we call it as one particular point. So point mutation also we can say. So because of this, what happened? This particular, I'm just giving an example. It is not that AGC going to produce phenylalanine hydroxylase. It's just an example. Okay. So this particular gene, okay, what happened? It will not produce this particular enzyme. Okay. So there will be no conversion of some factors that leads into multiple effect in the body, like a pigmentation, reduced pigmentation. Okay, that we are going to see here. So what will be the effect? Mental retardation. Okay, pigmentation, hair and skin color. It is not increased pigmentation, a reduced pigmentation. Okay, that's what a retardation we are given here. Okay, so these are the two effects. We are given only two effects. There are many other effects also. These are all the major effects like mental retardation and reduced pigmentation in hair and skin color. So why it is happening? It is happening because of one error. Okay, that error is because of that one particular gene. So multiple effect of a single gene is called pleiotrophy. Okay, so here it's a chromosome, the gene place might be here and the effect is seen here. So it leads into many effects. It's some other example. Eye, that may be change in the eye color, or there may be effect in the liver, okay, even in the heart, like that, right? So it is a flowchart representation. Pleiotrophy, feature 1, feature 2, feature 3 is nothing but the phenotypic features. Okay, one gene affects multiple characteristics. So before going into a detailed study about this phenylketonuria, I'll introduce about the chromosomes. In the beginning, introduction class, we saw something about chromosome. Okay, still more, these chromosome, how they are going to arrange and how they are going to find the defect that the particular person is having this type of defect, chromosomal abnormalities. Okay, that we will see. So it's a chromosome. We all know this image. Inside the chromosome, something is compressed. It is nothing but the DNA, right? And here it's a single chromosome, satellite chromosome structure. Okay. This we are going to see in molecular basis lesson in detail. This one I wanted to show. It is nothing but karyotyping, the arrangement of chromosome in this ideogram or karyogram. Okay. So how it is arranged? It is our human chromosome. See, it is arranged with the number. How many chromosomes we have? 23 pairs of chromosomes. 23 pairs of chromosomes, right? In the 23 pairs of chromosome, 22 autosomes, okay, that is body chromosomes. And one pair will be the sex chromosomes or allochromosomes, okay, allosomes we call it as. So this will indicate, okay, the sex of the individual, whether it's a male or female, right? And here the remaining all the body chromosomes will determine the body characters. Okay, if there is any effect here, okay, it will be shown like this, the picture. This is a colored version, and here you will have a black and white like a X-ray. and the if defect will be seen encircled, will be seen like this. Okay, so I'm just showing you an example how they will detect that the particular person is having some chromosomal abnormalities or any problem in the chromosome. Okay, so we'll go to phenylketonuria. This phenylketonuria, otherwise called as PKU, okay, in abbreviated form we can say it's an inborn error. And then, so in mutation, it's caused due to mutation where it is seen in chromosome number 12. For this only I showed how they have placed the chromosome in the ideogram. Okay, from that only they'll point out, okay, there is an error in chromosome number 12. And then in chromosome number 18, there is a defect. Like that, they can identify, right? So, low level of enzyme. What enzyme it is? Phenylalanine hydroxylase. That enzyme will be reduced. In turn, it leads into increased dietary phenylalanine. It's nothing but amino acid it is. So, what are the different? It is an autosomal recessive. Autosome just now is a body chromosome. A recessive, when they are in recessive condition only, the effect would be seen. Okay, both the gene should be, both the allele should be in recessive condition. For example, if we say small p, if they are like this only, it can be expressed. Okay. No phenotypic condition, heterozygous condition, it will be normal. Okay. Affects 1 in 12,000 babies. Treatment, this is the treatment that will low phenylalanine. There are two types like classic and variant. In classic, you can see lighter skin shade, and here some effect, smaller risk of brain damage also in variant PKU. Okay, and here the odor, smell, they smell musty. Normal health, normal lifespan as usual. If untreated, it leads into intellectual disability, seizures, fits, behavior problem, and mental disorder. Okay, how it can be diagnosed? By blood test. Okay, so next class we'll see about epistasis. Okay, the last topic of this different types of crosses, right? Thank you.